All right? So one thing about ALS is, unlike a lot of diseases, and especially cancers that are like one point mutation is what causes disease, that's not true of ALS. There's no one gene that's like, if I have this gene, I'm going to get ALS. We actually do have what they call an ALS gene, but ironically, only about 15% of patients have the ALS gene. And even people who have the ALS gene are not guaranteed to get ALS. So it's kind of odd. Only about 10% of cases are familial, which means that you know a family member that has ALS and you have an inherited variety. So 90% of cases are sporadic. And what's bad about sporadic diseases is that it means any number of things could have caused it, whether it's oxidative stress, the inability for the mitochondria to metabolize the free radicals and get rid of them, whether excitotoxicity is another theory where the nerves release too much glutamate and causes a toxic excitation of nerves that results in their death. It could be inflammatory, where you have astrocytes and microglia, which are the helper nerves, or helper cells around nerves that go bad, and therefore we don't really know. Sometimes the neuromuscular junction, which is where the nerve and the muscle connect, sometimes we think that the muscle goes bad first and causes that neuromuscular junction to disconnect, and in other cases it looks like the nerve retracts from the muscle so that it starts like in the spine or in the brain. So it's like all these crazy things happening with the biology, but yet they all result in the same phenotype that we see as ALS. The most common familial version of ALS is called the SOD1 G93A mutation. So that's superoxide dismutase 1 glycine 93 to alanine. So what SODs do, or superoxide dismutases, is they destroy free radicals, which is really important. You guys hear about cancer, and if you don't use sunscreen, all those things. Well, sun radiation is one form of energy that causes oxidative stress, right? So anyway, we have an SOD1 G93A mouse model of ALS. That's the #1 utilized experimental model for ALS. And even though it has this one mutation, and we know exactly what the mutation is, we still don't understand all the etiology. It's still multifactorial. There's all these things that are going on, both upstream and downstream in the cellular pathology. In the ALS mouse model, the high copy mouse, it lives about 150 days and has symptom onset at about 85 days. And this is when you look at a mouse's lifespan, that's similar to the onset and duration of what a human patient would be. And then what we use to measure the function of these mice or the disease progression is something called a rotarod, which is a spinning rod, and we time how long that they can stay on a rod that's moving at a certain speed, okay? We also can track their body weight because as they have problems swallowing, they, like humans, also lose weight. And then other things are like grip strength, like how well their front paw can grip something. So different functional measures. And then, of course, there's all sorts of biochemical markers that are done. All right? So that's my basic overview on ALS to kind of set you up for what you guys really came for, which is to hear about the computational aspects and the predictive medicine. All right? So the goal of predictive medicine is to, quite simply, expedite clinical success. We all know that FDA approval takes way too long, 7 to 14 years, and experiments also take a long time, and you can only look at one thing at a time, right? Very hard to do. So the goal of predictive medicine is to be able to expedite the identification of the underlying etiology from the subcellular to the cellular to the system level to predict new therapeutics, all right? And then also, let's not forget, we want to optimize existing interventions. One of my big beefs as a patient of a neurological disease is everybody's focused on making me walk. I would love to be able to walk. That would be awesome. But in the meantime, there's a lot of things that can be done to make my life better while we're waiting for that cure, right? So I feel like that for ALS patients. We may not be able to cure them, not for a year, 10 years, 20 years. I don't know how long it's going to take. But in the meantime, we can use predictive medicine to optimize the interventions that are available to make their quality of life better and to hopefully extend survival. All right, so there's a lot of talk about big data. That's really popular these days, and usually when you hear big data, you're thinking large file size or high resolution, meaning that you've measured 10 parameters 10 million times, right? And you're looking to make some sort of more simplistic model out of really large file sizes. Then there's also this thing called smart data, which is kind of the opposite. Instead of having a high number of sample sizes, you actually have a lot more parameters that you don't measure as many times, okay? So it's more about finding relationships in the data versus the reproducibility of the data, all right? It's very multidimensional and it's very much a dynamic system, all right? Usually big data, you're looking at what happens at a certain time point. You may look at different ones, but they're always segregated over time. It's not a continuous, usually, all right? So it's blood pressure at 8 a.m., blood pressure at 10 a.m., right? So smart data, we try to look at more continuous measurements and look at how those relationships drive the system. And my lab definitely leans more towards the smart data side, but you also have to have some degree of big data, all right? Big data usually encompasses these four areas. You have your electrophysiology and sensors, which is like your ECG, your EKG, all right, brain, heart, and the electrical signals. You have genomics, which is looking at that like glycine 93 to alanine, right? So we have specific mutations. We have imaging, where you're looking at like PET scans, MRI, X-rays, all right, and trying to like determine different stages of the cancer. And then you have proteomics, which is looking at how proteins fold and how they develop. And if something goes wrong, then of course proteins build up, right? So that's your traditional big data. Most people don't try to integrate all of those, all right? They just pick one, all right? So what my lab tries to do is not just pick one type of data, but rather integrate all types of data, apples and oranges, into what could be one comprehensive view of what a disease is. In this case, ALS. So I call this pathophysiology informatics. It differs from regular informatics, which is more focused on genomics, proteomics, what have you. All right, pathophysiology is multiscalar, so you have to look at it all. All right, so it's the application and integration of in vitro, that means inside the cell, in vivo, which is at the whole animal level, and in silico, which, as you guys know, is computational. All right, so you want all three aspects of that to get a true view of what's going on in a disease. And what makes this really different is that the data sets are scattered. All right, we don't have the luxury of doing one experiment where we got 10,000 samples. Instead, all the data is scattered, and usually across different publications, right? Somebody published in Science, somebody published in Nature, somebody published in ALS FTD. And so all these data sets, they're not one database. They're all over the place. And so you've got to be able to get them together so that you can get a big view. The other thing is it's disparately expressed. How a protein person measures their data compared to how a rotorod is measured compared to how a gene is measured. They have very different units of expression. And then besides that, you've got different protocols, right? And so how do you deal with all these differences in the data? So this is what pathophysiology informatics is about. So we need to access, aggregate, and analyze all these disparate pools of data. So I kind of look at this as like a quilt. If you imagine every time you publish in a journal, you're contributing to a patch on a quilt, all right? Inside your patch, you may know all about what goes on in your patch, but it's really stitching all those patches together that gives you a true view of what the disease looks like. So that's kind of like what our goal is in the lab, and specifically with ALS. I call this viewpoint aggregation. You're looking at all the different viewpoints from experimental, clinical, and theoretical to build this quilt. And the more patches of the quilt we put in, the higher the resolution the quilt. The better the resolution, the more we know what's going on, right? So eventually, if you get enough patches inserted, you can see a pattern that you wouldn't see if you were only looking at your one little patch. All right? So the idea of this quilt theory is based upon complex theory. And complex theory really focuses on the relationships between local subsystems or local parts. You may have heard of the word emergence or emergent properties. So, for example, the calcium and sodium channels inside a neuron work together to produce an action potential. No one sodium potassium, whatever channel can make an action potential, but they all work together, and we have this emergent thing called an action potential, all right? So in complex systems theory, you have dimensional restriction and local relationships. And it's called a bow tie because you have all this information coming in, but it is dimensionally restricted based upon how the relationships dictate. And then outwardly, we have this complex adaptive behavior. And then from an engineering sense, we have amplification and dampening, okay, in order to kind of turn up or turn down. It's like a radio knob. How high is the volume, right? And of course, in a biological system, we want homeostasis, which means that based upon what input we get, we may have to turn something up or down in order to get that homeostasis. All right, and so if you're more interested in what this complex theory stuff looks like, a good paper I recommend is this Sethi and Doyle paper in Trends in Biotechnology, published in 2004. And it talks about it using metabolism more at a metabolic stage, but this applies to all of biology. Okay. All right. So I just mentioned that system stability and homeostasis is everything in biology. If you don't have homeostasis, then you're sick or you're dead. All right? So one thing that we like to consider is, what if disease is really just a phase of instability? In other words, if we could just restabilize the system, we could fix the disease. And if we can restabilize the system, we don't actually have to know what the initial perturbation was. We just have to restabilize the system. That's really nice, because in case like ALS, where you have all these different causes, you don't want a treatment to target every individual cause. You just want to fix the phenotype, which is ALS. So one thing we all have in common is our compensatory mechanisms. So if we can use the idea of stability, just like a little plant that you would see in chemical engineering, gains and those types of things, we can make a comprehensive model to see the instability, and does that instability correlate with disease progression? All right. The method that we utilize to do this, to develop a system model, all right, and it's kind of cute. That way you'll remember, right? AAA: access, aggregate, and analyze. You got to access the data, all these disparate data sets. You got to be able to aggregate apples and oranges, and then you got to pick the right tool in order to analyze, because there's not a one-size-fits-all analytical approach. All right? So here's my little army that Ashley and I have been talking about. All right? So we have a lot of people working in the lab. All right? They're mostly undergraduates, although I do have some graduate students too now. We take a lot of people in, 35 to 50 a semester. All right? They learn how to access this data, which is mostly in publications, but we also access clinical data. They're taught about ALS, the basic biochemical principles that I can teach them, the ones we know of, a little bit about data science. Then they take an exam. Then they learn how to curate stuff, whether it's, publications or power chart, whatever it is. If it's clinical data, it's experimental data. We have a live curation server where 20 people can enter data at the same time into a database. Then we have quality control, because you can't have anything in engineering unless you have quality control. And everybody has to look at what's been entered at least 3 times to ensure that we have 99% accuracy or more. And then we have our analytical teams, which are usually 3 to 4, maybe 5 people who work together on these little pilot projects, whether it's one patch of the quilt or whether it's doing the stability analysis on the whole quilt. Okay? And then we have management, which you have to have oversight when you have so many people. And they're like their own little corporation, and it works really well. So if you ever want to set one of these up, let me know, and I'll tell you everything I know about it, all right? One thing I do know is it's been very successful, and I'm not tooting my own horn here. I'm very proud of my students, and they've done very well. But people in the lab, I said a 3-month tenure. They should have had 3-semester tenure. So if they stay in my lab 3 semesters to do this work, it was equivalent to a 0.6 GPA boost in terms of their competitiveness for jobs, all right, or to go to medical school or graduate school. They've done very well, more than 85% acceptance. Last year, I think I had 14 apply to medical school, and they all got in. I was very happy. They've done really well. Lots of first authors on publications. It's great. So if you want to set one of these systems up in your lab, I think Oklahoma State has some talented students, and I think they would do really well with a system like this. And we won an award for it. All right, so that's the, we'll talk about the access piece a little bit more here.